Hello, I'm Yichun. I'm a master's student from University of Florida. The project of my poster is about epigenetic regulation of a transcription factor NSP2, which controls peanut nodulation. Peanut is an allotetraploid species with AABB2 subgenome. As a member of the legume family, peanut has the capacity of nodule formation to perform biological nitrogen fixation. We have identified that NSP2 encodes a transcription factor controlling peanut nodulation. The wild-type NSP2 results in NOD+ phenotype, and it is dominant to the mutant NSP2, which exhibited NOD- phenotype. Interestingly, NSP2 of A subgenome follows Mendelian inheritance, and the separation rate of F1 heterozygotes is 3:1. While the NSP2 in B subgenome exhibit no Mendelian inheritance pattern, the separation rate is nearly 1:1. That's because the heterozygous NSP2, big B, small b, are either NOD+ or NOD- phenotype. The results of real-time PCR indicates that the NOD+ plants only express the big B allele. Similarly, the NOD- plant only express small b allele. Based on the result of reciprocal cross, it seems like only the male gametes contribute to the next generation's phenotype. Genome imprinting is an epigenetic phenomenon which leads to the parent-of-origin specific expression. DNA methylation as the epigenetic mark imprinted in the germline cell of parents to suppress gene expression. In this case, we hypothesize that the maternal NSP2 gene is imprinted then silenced through DNA methylation. For the first goal, we aim at to compare methylation variation at NSP2 locus between the male and female gametes. The methylation status has been revealed by biosulfite sequencing. 40 flowers, pollen, and ovaries were collected from each individual plant. Meanwhile, the leaf and peck tissue were also collected as control. Pollen and ovary DNA were extracted following by biosulfite conversion, which can transform the unmethylated cytosine to uracil, then convert it to thymine, whereas the methylated cytosine still maintained to cytosine. As the DNA double strand cannot pair anymore, the biosulfite converted DNA are highly fragmented and show smear on agarose gel. The amplicon size are relatively short due to the integrity of biosulfite converted DNA. And due to the highly similarity between A and B subgenome, only a few sites could be selected to design primer. Overall, there are 12 pairs of primer were designed to cover the promoter and CDS region of NSP2B. The biosulfite PCR were performed, and the PCR products were sequenced by Sanger method. After comparing the biosulfite sequencing results and the original sequence, we identified the methylated pattern of NSP2 locus in leaf. The total methylation rate is 15.87%, and the partially methylated rate is 14.09%. For the ovary and pollen samples, different methylation region between different plant tissue with the same genotype appear from 22 to 217 base pair. In general, pollen and leaf contain small methylation sites were observed than ovary, particularly at the B region. Leaf has both methylation and unmethylation at this loci. Gametes show either methylation or unmethylation. Only pollen exhibited methylation on some specific loci. It may be explained as leaf contains both male and female alleles, whereas the male or female gametes only occupy single allele. In the future, we are going to perform the same process to more tissue and biological replicates. Next, we are going to perform qRT-PCR to reveal the NSP2 expression level in male and female gametes. Well, thanks for listening. If you have any further questions, please feel free to contact me.